Alright, let's cut to the chase. You want to know how ELISA works? Think of it as a tiny, super precise Avengers-level detective for proteins. What's the basic idea? We use antibodies as the sniffer dogs to find a specific antigen, or vice versa, then convert that binding event into a measurable signal. So what are the main flavors of ELISA and why choose one over another? There's direct ELISA, slap your antigen on a plate, add an enzyme-linked antibody that recognizes it, and read the signal. Fast, simple, but sometimes not sensitive enough. Then indirect ELISA, antigen on the plate, primary antibody from your sample, and an enzyme-linked secondary antibody that recognizes the primary. More sensitive because signal amplifies, but introduces extra steps. Sandwich ELISA, capture antibody coats the plate. You add sample so the antigen is sandwiched between capture and detection antibodies. Extremely specific and great for complex samples. Competitive ELISA flips things around. Sample antigen competes with labeled antigen for antibody binding, useful for small molecules or when only one antibody is available. What are the step-by-step essentials? You coat a microplate well with capture antigen or antibody. Block non-specific sites so nothing else sticks. Add your sample so the target can bind. Wash away the rest. Add a detection antibody linked to an enzyme, like HRP. Wash again, add substrate. Enzyme converts substrate to a colored or luminescent product, then measure absorbance or luminescence. Controls. Critical. Positive, negative blanks and standards for quantification. Why does sensitivity vary? Factors include antibody affinity, signal amplification strategy, blocking efficiency, and sample matrix interference. How can we make it better? Use high-affinity antibodies, amplification systems, optimize blocking and wash conditions, or switch to luminescent substrates for a wider dynamic range. Beyond the lab bench, what should we ask about ELISA's role in society? How do we balance accessibility and reliability when tests affect public health decisions? Could we miniaturize and automate ELISA even further to make rapid, at-home diagnostics without sacrificing accuracy? And if antibodies are our detectives, how do we ensure they aren't biased by cross-reactivity, leading to false leads?
Bottom line, ELISA is a versatile, powerful technique for detecting and quantifying proteins and small molecules. It's part engineering, part chemistry, part detective work. Want me to design a hypothetical rapid ELISA device for your smartphone next?